Then coming to phase one. So from phase one onwards, these are the compulsory phases that we cannot skip on any of these phases. So if we want to do the trial, we have to start with phase one. Phase one is also known as human pharmacology or human or clinical pharmacology studies. So these are the studies that is the phase one is the phase in which the humans are first time involved. So this is known as first human study. So the question that is being asked as is that in which phase we first time start the human trial. So whether it's phase 0, phase 1, phase 2, phase 3. So the answer is phase 1, not phase 0, because phase 0 is not compulsory. So in phase 1, we take healthy people. In phase 1, the main objective is to check the safety. So once we have checked the safety in animals, then we are going to again confirm the safety in human by doing the phase 1 trial. And in phase 1, we again check the safety of the drug in the healthy people. Okay, so phase 1, we don't take the diseased people, we take the healthy people, because if in case, suppose we want to discover the medicine for hypertension, for high blood pressure, we want to find out a new medicine. So if that product or that molecule is being tested in the person who is suffering from high blood pressure, if that drug is being given to that person, and if the drug is going to show the side effect, maybe the condition of the person get deteriorated and the patient becomes more serious, more sick, and maybe because of more rise in blood pressure, the patient gets the stroke. So that's why in phase 0, as long as we are not sure about the safety of the product, we are not going to give this to any diseased patient because it can cause more severe reactions in them. So if we give the drug of hypertension drug to the healthy people who are not having the blood pressure, so if we give them, and we know that all their body parts, all their organs are working well. So if they consume the drug and if the drug is not effective or if the drug is going to show its side effect, so those side effects won't be so severe for the healthy volunteers because those side effects with the help of treatment can be managed. Okay, so the damage to the healthy people won't be much in comparison to the disease people. Because diseased people already their organs are not working well, and if we give them some drug that is not safe for them, can deteriorate their condition and can lead them to more severe reactions. Okay, so that's why in phase 1 we take the healthy people, we don't take the diseased people. But there is an exception. The exception is cancer and HIV. If the drug is being discovered for HIV or for cancer disease, then in that phase, we don't take the healthy people. So in every trials, we take phase 1, we will be considering the healthy people. But if we talk about the cancer or HIV, in these two conditions, we are going to take the disease patients. Means the cancer patients will be taken in phase 1 as well. Why? Because these medications that are being used or these therapies that were being given for the treatment of cancer or for the treatment of HIV, so these can cause a severe illness in the healthy people. So these drugs are more toxic in nature and can cause a huge impact or damage to the healthy people. So that's why the cancer and HIV drugs are not given to healthy people. The cancer and HIV drug will directly be administered to the HIV and cancer patients. Okay, so the main objective of doing phase 1 is to find out the safety of the drug plus to find out the maximum tolerated dose, that is MTD. MTD is the maximum tolerated dose, but maximum dose that can be given safely without causing unacceptable side effects. That dose is known as MTD. So what we do in phase zero, we form groups. We suppose we have created 10 groups of 10 people. Each group contains 10 people. And we started with maybe 25 mgs being given to group one. We found that the drug is safe. We moved to group two. Group two is being administered with 50 mg and it is found to be safe. Then we moved to group three and we gave it around 100 mg. Still, the drug is found to be safe. Again, for group four, we just gave it maybe 250 mg. It's found to be safe. So, in the phase one, what we keep on doing is we keep on increasing or we keep on escalating the dose. So the dose will be escalated or the dose will be increased till the time point we feel that it is not going to give unacceptable side effects. So the moment we come to know that the drug is causing severe side effects, that is the point when we will stop the drug. And we will say that this is the maximum dose that will be tolerated by the human body safely without unacceptable side effect. Means the side effect will be there with the drug. So no drug is available in the market without side effect. So every drug has the side effect, maybe minor side effect, but it is there. So those side effects are acceptable one. Okay, so the drugs that we take, suppose we take any medication, maybe for fever. So we know that if we are taking paracetamol, it is not good for liver. We know that. So that is one of the side effect of the paracetamol, that it causes some or the other damage to the liver. But that the damage is very minor, so that is acceptable. But if the person is already having the disease, liver condition, and if that person is being given that drug, it will be toxic for them. So that's why when we say that the unacceptable level of risk means the side effects are acceptable. Maybe after taking the drug, the patient side effect is constipation or the patient feel nausea or vomiting. So these are the side effects that is accepted. That if you take this drug and your our condition is getting better, but it's the causing the side effect. It's just like you get that feeling of nausea or something. So these are acceptable side effects. Unacceptable side effects are the which can lead the patient in a serious condition. That after taking the drug, the patient's condition becomes serious. A patient has some stroke, or the patient have severe uh, any other disease condition which is not acceptable. Okay, so that is what we try to find out in phase one. So in phase one, we check the safety of the drug and we try to find out the MTD, that is maximum tolerated dose. So maximum tolerated dose is the maximum dose that can be given to the patient without having unacceptable side effects. So maybe the 650 is the maximum dose that can be given with acceptable level of side effects. Okay. So side effects that the drug is causing is acceptable in terms of the benefits that the product is going to give. Plus the pharmacokinetic and pharmacodynamic profile will be checked in all the phases. Is this clear?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
So phase one is further divided into SAD and MAD. So the first step is SAD, and once the SAD is completed, we move to MAD. So let's understand what is SAD and MAD. So SAD is a single ascending dose. So this is what I have already told you, that we form small groups and small group is given a particular dose of drug. Okay, and those people, those patients are monitored and this drug is being given maybe for a week time or for a 15 days time or for a month time, and we observe the patients whether they are having any side effects or not. If no side effect, we escalate the dose, means we increase the dose in new group, means group 2 with a higher dose, maybe 50 mg. And this step by step, we keep on increasing the dose of drug in new group of people. And we will continue this till we find the dose which is maximum acceptable and which is not going to give severe side effects. So the highest dose with acceptable side effect we are going to find in the SAT, that is multiple, that is single ascending dose. In single ascending dose, we are going to find the maximum tolerated dose. Okay. So that is what we are going to do. Inside, we take small dose and we keep on moving in the ascending order till we get the maximum dose which is safely given to the patient. So once the SAT is completed, we will move to the next step or next phase, that is MAT. So MAT is multiple ascending dose. So here single ascending dose means one group of patients will take one dose a day and we keep on increasing the dose. Okay. Multiple ascending means the patient will receive more than one dose in a day. Means the patient may consume 2 times a day the drug. Maybe in the morning and in the evening, the patient is being given a dose of drug. So the process will be same. Group 1 will be given 25 mg in the morning and 25 mg at the night. Then the second group will be given 50 mg in the morning and 50 mg in the night. So this will be continued, and we are, after giving the drugs, after 1 hour or 2 hours, we are going to collect the blood samples from the patients and we are going to do the analysis. So what we exactly wanted to find out after collecting the samples, that how long the drug stays in the body, how long it takes for the drug to be completely processed in the body and to be eliminated out of the body. So accordingly, the doctors prescribe the dosing. Some drugs can be just taken once a day. Some drugs can be taken twice a day. Some drugs can be taken thrice a day. Some drugs can even be taken 4 times a day. Means an interval of 6 hours the drug can be given. So it completely depends upon the elimination time of the drug. So how long the drug stays in the body. If the drug stays in the body for 12 hours, the second dose will be given after 12 hours. If the drug stays in the body for 24 hours, the second dose will be given after 24 hours. And if the drug stays in the body for 6 hours, the second dose can be given after 6 hours. So this is on the PK parameter of the drug. We decide that when, how frequently the drug will be given and what should be the dose of the drug in a day that can be given safely to the patient. Yes, so one paracetamol we know can be given after 6 hours, and giving 4 doses of paracetamol in a day is completely safe. Okay. And if some drugs takes around maybe 12 hours of time and we give the dose of the drug after 6 hours, so the drug is not completely eliminated, the drug is still in the body and we have given the second dose of the drug. So there will be the chances of overdosing of the drug, which can make the patient's condition maybe more fatal. So that's why it is important to know that when the second dose of the drug should be given safely and how many doses in a day is safe for the patients and how long the drug stays in the body. So mainly the pharmacokinetic profile will be checked here, and we are going to determine the dosing, frequency of dosing, how many doses a day is safe for the patient. That we are going to check in the MAD. And SAD, we are going to check the maximum dose that is being given. And here we are going to check maximum dose, how, what interval should be given. So once a day is safe, twice a day is safe, thrice a day is safe, thrice a day is safe, yes, and 4 times a day is safe. Yes. Is it clear?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes.